Bismillahirrahmanirrahim. We talked about the basic structure of bacteria. See, bacteria are unicellular, right? They're only one cell. You'll never see a multicellular bacteria that's mutant bacteria. They're unicellular, like one cell at a time. All cells must have a cell membrane. It's like a boundary. It's like how can you have a house without walls? You know? It's like that. A membrane is like the walls of a cell. Every cell in the world has to have a membrane. It's like you can't have a house without walls. Basic is that. Bacteria, in addition to the cell membrane, have a cell wall because of their peculiar needs. They have a circular DNA. The mush inside is called the cytoplasm. They have ribosomes. They make proteins. Some bacteria have capsule. The analogy I gave you was that if the membrane and the cell wall were like a French fry, if you dip the French fry, put that dip in ketchup, that's a capsule. That's additional material. And then we talked about the difference between gram-positive and gram-negative bacteria. Some additional details of bacterial structure. Now, these things called plasmids. We'll get to know plasmids really good someday. There'll be like a whole talk about plasmids, okay? Plasmids are like small pieces of DNA which have only a few genes. In addition to the big circular DNA, it has some small pieces of circular DNA. So bacteria can have one plasmid or it can have multiple plasmids. Now, why have these extra plasmids, huh? Many times these plasmids are genes that the bacteria uses a lot. And some bacteria, now, all bacteria have cell membrane, all bacteria have cell wall, all bacteria have circular DNA, all bacteria have ribosome. Not all of the bacteria have plasmids, not all bacteria have pili. Pili are these little projection things, okay? And not all bacteria have this tail. It's called a flagellum. It allows them mobility. Pili are basically, you know those darts you shoot dart game, and then it goes to hits the thing and there are numbers on it. Dart game, that the circular balls they have the sticky thing on. That's like a pili, the Velcro type thing. That's a pili. That's exactly the function of pili. Because if you take a ping pong ball and hit it on it, it's not going to stick, is it? So pili are like Velcro material for bacteria. They allow them to stick to any surface. Now those thingies that you throw on that thing, right? They'll stick to anything that's not smooth, right? Like your throat or something. Yeah. So they have pili. Also, they're like ready-made tunnels. So two bacteria, it's fascinating. It's not that bacteria, they don't communicate with each other, right? It's like one cell, they're unicellular. It doesn't mean that they don't communicate with other bacteria. Oh, they do. They do. Bacteria, you'll see in a second, they even mate through pili. They're like tunnels. And the bacteria comes over, the tunnel connects, and the two bacteria are connecting. See, when people mate, when people have intimate relations, when people mate, what the biological purpose of that is exchange of genetic material. Yes or no? Yeah? That's the biological purpose of mating. So these two bacteria come together, pili grows in a tunnel, right? And then there's another bacteria, and they exchange DNA material. These little plasmid things goes from one bacteria to another bacteria. Now that other bacteria has the genetic material that it did not have before. That's why it's called bacterial mating. So we talked about this green thingy here, which is right here, plasma membrane. Outside is the cell wall. Outside is at the capsule. These are cartoon pictures, right? We talked about the circular DNA. All clumped up, it's called the nucleoid. Nucleus. It does not have a nucleus. No nucleus in bacteria. Right? Because they're prokaryotes. If it had a membrane around it, that would be nucleus. But no nucleus. Haram for prokaryotes. Yes. So nucleoid means nucleus-like area. Ribosomes, pili, you got the pili, the plasmids. Okay. On the right side, you see all bacteria have those structures. On the left side, sometimes they have that structures. So all cells must have a cell membrane. All bacteria have cell wall. All have chromosome, the circular DNA. All have ribosomes. Sometimes they have plasmids, sometimes they have capsules, sometimes they have flagella, right? Now this is identifying bacteria. Gram-negative bacteria. Now how do we figure this out? See, if you take your sputum, it's my curiosity. Watch this, okay? Let's say if you had a microscope, you put the sputum on the microscope, right? And you just look inside there. You know what you see? What do you think you see? What? Nothing. Because things are so small, they're transparent. One cell, ordinary, is transparent. It's so thin. It's tiny. Yes? So you look at it, you don't see anything. But then one day you get curious. Because, there's ink over there. There's spit over here. I wonder. Right? And there's blue ink and there's red ink, right? I wonder. So blue ink, red ink. I wonder what happens if I put blue ink, red ink. So you put blue ink over there. You put red ink over, you start seeing things. Look, you know what paper is? What's paper? Where does it come from? Trees. What part of trees? Inside a dead tree, right? That part. You know if you wash everything away from the tree, you know what's left? The cell wall. You know what this is right here? Cell wall. Do you write on cell wall? Simple as that. Yes? The paper comes from trees, yes or no? Trees are living things, yes or no? It's cell wall. It's processed cell wall. So, does ink stain the paper? Of course it does. Same principle. You're writing on it like paper. You're writing on cell wall. Think about that next time. So, bacteria are identified according to gram stains. Different dye taken up by different cell walls. Cell wall is made of interlocking sugars and amino acids, as we talked about. The proteoglycans. So different types of cell walls take up different colors. There's certain type of bacteria that love blue-violet color, and there's certain type of bacteria that love red color. The ones that are red color taking, they're called negative. The ones that are blue, they're called positive. So this gram-positive, it's kind of easy to remember. Right? What do you think about the negative? Red, like blood, yeah. Gram-negatives are red, gram-positives are blue. How do you do it? Well, you take spit or whatever you're trying to stain, right? And you put it on the slide, and then you put violet, just basically ink on it, and then you put iodine, which allows it to stick to it. And then you wash it because you don't want too much of it. If you didn't wash the iodine, you put it back, everything looks blue because you're just looking at violet colors. Like, oh, it looks like ink. It is ink because you didn't wash away the excess ink. Yeah? So you put the stain and you wash the excess stain, so you're not looking at the ink, you're looking at the bacteria. And then you apply the saffron, the pink stain, and then you look in there and you enjoy it. That's one way. So how do you identify bacteria? Why is it important when you identify bacteria? Very important. Why do you think they take patient's blood and try to grow the bacteria in the petri dish? And then once you grow the bacteria, and then you stain the bacteria and look at it in the microscope, aha! We have gram-positive bacteria. Should we tell the doctor? Nah, don't worry about it. No. Why tell the doctor? Why tell the doctor? Because the antibiotic that kills the gram-positive bacteria is not the same antibiotic that kills the gram-negative bacteria. You want to give the right antibiotic to the right bacteria. Do you understand? That's why. Okay? So it's important to identify. Yeah, but who cares? You just want the cell wall. In fact, when you put the bacteria over there, first you go like this under the Bunsen burner, heat it up. Heat. So to stick it. If you put spit on the thing and you heat it, water evaporates, or what's left over is organic material. So the first thing you do is you kill the bacteria in a way. It may not kill all bacteria. You understand what I'm saying? You don't care as long as the cell walls are there, right? You don't want to know what kind of DNA it has. Yeah. Okay. So one way of identifying bacteria is by what kind of stain they capture. The second way of identifying bacteria is how they look under the microscope. Because some of them look like this. They're still called spirochetes or spiral bacteria. Like syphilis, it's a sexually transmitted disease, STD, syphilis, is a spirochete like that. The more common ones are they look like that. They're called bacilli. And then these are called cocci. Some people call it cocci. Most people call it cocci. Like Staphylococcus aureus, skin infection. Bacilli, these are the common shapes. Bacilli, cocci. These are obviously not nearly as common. So these are cocci, bacilli. These are spiral bacteria. Now this is like clustered. There's a whole bunch of them here. You see, there's this cluster. There's like, over here, probably 50 of them. This is one bacteria. If they look like this, they're called cocci, like Staphylococcus aureus. They have staph infection. Streptococcus, strep infection. That's strep. Strep throat. Bacteria looks like this. Coccibacillus, meaning somewhere in between. A little bit of this, a little bit of this. It's not exactly like spherical. It's not exactly long. It's a spherical long. And this is Vibrio. It's comma-shaped. Famous cholera. Vibrio cholera. That's its name. Diarrheal illness. Very preventable. There are two types of reproduction. Okay, reproduction is generation of the next generation. Right? There are two types of reproduction. Sexual reproduction, asexual reproduction. Now, what's the difference? You should know the difference now because I just explained it to you. What do you think? Okay, I'll give you the words because you're probably shy of saying the word. Okay, in sexual reproduction, what happens? No, people, you call them bacteria, people? No, to organisms. Sexual reproduction means there's exchange of genetic material. So if something reproduces without exchange of genetic material, that's asexual reproduction. Asexual reproduction, no exchange of genetic material. Sexual reproduction, exchange of genetic material. So bacteria do both things. Bacterial asexual reproduction is called binary fission. Binary means the two. Fission means dividing into half, basically what that means, okay? Dividing into two. Bacteria, see, they don't have no nucleus, all right? So what happens is the circular DNA replicates itself. So it makes two, one out of two, you'll see, okay? One circular DNA becomes two circular DNA. And the two circular DNA separate into two ends of the bacteria. And then the bacteria cell wall divides into two like this. Is there any genetic material exchange? No. So this is asexual reproduction of bacteria. I said one of the characteristics of all living things is that they grow, right? Yes? Okay. When the bacteria divide into two bacteria, the new bacteria are small compared to their adult size. He's just a teenager. He was born 5 minutes ago. So, yeah, they grow. The new bacteria is smaller. This is binary fission. There's no exchange of genetic material. This is real. These are bacteria dividing. They're all dividing by binary fission. Just watch. And if you observe carefully, the new bacteria is smaller than the parent bacteria. And then they grow. If you just try keep an eye on one of them or something. Yeah, the new ones are smaller. Does that make sense? They grow. Bacteria divide this fast. How come they don't take over the world, right? How come they don't take over the world? Some of them divide every 20 minutes, right? How come? Well, it's like this. We grow bacteria in a petri dish, right? Now, who can tell me why we use the petri dish? It has food. So the petri dish that over there, okay, it's not empty. It has lunch. It has food for the bacteria. Let's say if you put, some specimen, let's say from your hand or from your shoe, put it over there. Next to you come is going to have lots of bacteria, right? Let's say you sample somebody's wound and then you put on a petri dish and nothing happens the next day. What does that mean? They didn't like the food. So some bacteria like biryani. You better give them biryani. They ain't going to go nowhere. That's why you have different types of petri dishes. they have different nutritional requirements. So you don't take a swab of somebody's wound and put on one type of petri dish. You put on 5 different type of petri dishes because they have different nutritional requirements. Does that make sense? Okay, that's one thing. So a petri dish has food for bacteria. Then you say, how come the bacteria just don't go eat up the petri dish and go out of the petri dish and all over the table? How come they don't do that? Because there's not enough food for everybody. So when the food is done, they stop dividing. So why don't bacteria take all the oil, thank God? Because there's not enough food for all of them. They immediately win, right? Then they also make poisons of their own. They limit their own growth. And they're predators. They're stuff that eat bacteria. There are limiting factors that limit the growth of bacteria. Got it? This is sexual reproduction in bacteria, which means what? Exchange of genetic material. Do you see the pili? See, what happens here is there are 2 bacteria. The pili connect. And then the plasmid over here duplicates itself and gives the plasmid to one of the plasmid copies goes to the other bacteria. Now it has the plasmid that it didn't before. Okay, look what's going to happen over here. It's a demo. Okay, I just want to tell you something, right? Watch. It's like big subjects, like big words, right? Follow? But there's a way of doing it in a way that's very decent, right? We have to always watch that. The next slide that you're watching, it probably took me a half hour to make that one slide. Right? When I make these slides, I want to make sure that everything's proper. You know, I clean it up if I have to, things like that. Because it is important. This is what we have to bring to this culture. Because if you were to Google something like this, on Google, sexual reproduction in bacteria, if you go in a video, and if it's a live video, half the kids in the class will be laughing and making jokes. Because Allah has not gifted them with Allah has gifted you. Don't forget that. These are important things. The Messenger of Allah came to refine our character, our akhlaq. Now, that is so important, beautiful kids. Listen to me, that is so important. Then you are the next generation of teachers. You understand? You have to take that message that there's a way of learning that shows who we are as people. So bacteria, when they come together to reproduce, that's called conjugation. Conjugation means coming together. Coming together means conjugation. So the pili goes and connects to the pili of the bacteria, right? And then they exchange genetic material. So look, there's the exchange of genetic material. There we go. Do you see that? That's sexual reproduction in bacteria. Now this is how it happens. See? Pili go. You give antibiotics to the patient, that antibiotic works. Okay? Patient gets better. And after a while, the patient gets sick again. Because you killed all the bacteria that the antibiotic could kill. And then there's some left over that had the plasmid that has antibiotic resistance. And the one that had the antibiotic resistance, right, flourished despite the antibiotic. Does that make sense? Now somebody else gets infected and has its cousins over there. They do one of those pili numbers, you know what I'm saying? They exchange the antibiotic resistance. So now you have an epidemic, which is a bacteria that's very difficult to kill. So genetic exchange occurs in bacteria. Okay, so there are 2 ways that bacteria divide. The binary fission is their asexual reproduction, and we talk about conjugation, which means coming together. Adaptations in bacteria. So they're anaerobic bacteria, possibly the first photosynthetic organisms ever. Now they're aerobes. Aerobes mean they like oxygen. Aerobic exercises. So aerobes are bacteria that are okay with oxygen. And there's certain bacteria called obligate aerobes. They must have oxygen. Like Mycobacterium tuberculosis. That's why it's going to go and infect the lung. Okay? because it must have oxygen. And then they're obligate anaerobes, means they're killed by oxygen. Obligate is must necessity, like syphilis, Treponema pallidum, syphilis bug, etc. Botulinum, they're killed by oxygen. So, and then they're either they can be aerobic or anaerobic, depending on circumstances. They use the oxygen for the same purpose that we use oxygen for their breaking down of foods. Endospore formation for survival. Okay, when the going gets tough, this kill the bacteria, but you can't kill the spores. It's a hibernating mode. So a spore is the tiny structure of the bacterial DNA. Okay, now look. If bacteria says, okay, antibiotic coming, I better pack up my bags and make a spore. You know, what's the most important part of the bacteria, you think? DNA. So if it's going to pack up, what is it going to pack up? DNA. So it takes the DNA in one area, and it takes the cell membrane, right? It makes like cell membrane, right around it, right? So you have, DNA inside of it, and there, that much cell membrane. And on top of that, cell wall. That's a spore. So here's a bacteria that's going to make a spore, see? And the spores looks like this. So here's a bacteria, the same bacteria in the process of making a spore like that. And that's a spore, that's mature spore. It gets rid of everything else. Does that make sense? Watch, it's going to make a spore. Okay, just watch this over here, okay? Cell membrane. More cell membrane. This is a cartoon picture, so it's actually many more cell membrane. Like, you follow? There we go. See? And then this one tells you how it comes back to life again. That's the spore. Ta-da. It's like unpacking. I'm home, honey. This is like useful information. The reason you have stuff in cans that have been like that is to prevent endospore formation. If you ever see that and you see a little film of stuff on top of it, throw it away. So canned food, so to kill endospore, you sterilize, heat it under pressure, and can the food, sterilize and acidify. If you don't do all of the process, so this is all the clean stuff, endospores all killed. Okay, once you open it, you have a possibility of getting endospores again. Clostridium botulinum, Clostridium difficile. Okay, two different species, same genes. So this one also makes spores, right? So if you see swollen dented cans, don't do it. Off order, don't use it. If you reheat stuff, make sure you really reheat stuff. Botulinum toxin, it's a nerve toxin. So they get double vision because one eye is looking this way, other eye is looking this way, because the muscles of the eyes don't function in tandem. Okay, so they have difficulty speaking because the muscles don't work. They have difficulty breathing, they're in trouble. They love this environment, so don't store a hot potato because they love the metal there. This was anthrax, anthrax. And if humans inhale anthrax, they can get lung disease and it can be fatal. Importance of bacteria, we talked about nitrogen fixation. That should be pretty clear to you guys. Bacteria, along with fungi, are major decomposers. Bacterial diseases we talked about, most of them harmless. They come through skin or wounds, or we eat them. Diseases occur because whatever's causing disease interferes with the normal functioning of that organ. This is tuberculosis. It starts normal lung, eats up the lung basically. The biggest disease today is not leprosy or tuberculosis, but rather the feeling of being unwanted. I wonder if this child like this, I wonder, aren't there rich people in the world? So this is Alexander Fleming. So he did this, he was trying to grow this bacteria, Staphylococcus aureus bacteria, okay? So he did his thing with the petri dish, okay? And then he came in the next day, He looked at the petri dish and he saw the bacteria, but then he saw a fungus. And he was going to throw it away. But that would not be scientific, because he observed something. Seeing is not the same thing as observing, right? What he observed, as you can see over here, he saw the bacteria grow, and then he saw a fungus colony, but what he noticed is there's no bacteria around the fungus. It's like, don't come here! Yeah? How come the bacteria just didn't keep growing up all the way up to the fungus? He reasoned that the fungus was producing something that was inhibiting the bacteria. Okay, the name of this fungus is Penicillium notatum. The thing that was inhibiting growth is named after the fungus, and it is penicillin. He tried in his life to find what chemical it was that was inhibiting the growth of that bacteria. He didn't do it. It happened like a decade later. Okay? But he's credited for it. Why is he credited for? For observation. He discovered that. And then eventually the penicillin was isolated just before World War II. Helped save millions of lives. And that's it. As-salatu was-salamu ala rasulillah wa alhamdulillahi rabbil alameen. Assalamu alaikum.